She is a researcher also here at PNNL with me. We work together on a lot of projects. And she's going to be giving a talk this morning, focused again, like on the metagenomics that we've been seeing. But while Susanna was talking a lot about the maybe bacterial or microbial contributions to what's happening in these microbial systems, Ruonan's going to talk a little bit about that, but also kind of take a step away and maybe look at where other genes might be coming from outside of microbial or bacterial or fungal systems, maybe even phage and viruses. So Ruonan, did you want to go ahead and share your screen and take it away?
So hello, everyone, and thanks, Ryan, for the introduction. And yes, I'm Ruonan Wu, bioinformatician from PNL with expertise on genomic analysis, multi-omic data integration, and particularly interested in studying viral ecology. So that's why today I'm going to talk about how to detect viral signals from metagenomes, as it will heavily rely on the current bioinformatic tools and methods. So there will be some remaining limitations or challenges that require us to interpret the result with caution. So today I will walk you through the basic steps, review tools or methods to use, then highlight potential challenges and limitations for each step, and then suggest mitigation plans. And also due to the time limit, I probably won't be able to provide detailed commands or scripts for you to run each of these tools, but hopefully can provide you with a comprehensive overview of how to study viruses using bulk metagenomes. Then before we jump into the details, let me try to use one slide to get you excited about studying environmental viruses so we can still stay motivated when we go through the details later. So viruses are everywhere and abundant. So as shown in this figure from a global virus survey, that viruses can be detected across a wide range of ecosystems, including freshwater, marine, and soil, etc. And also they are quite abundant. So it is estimated up to 10 to the power 9 to 10 viral-like particles per gram of soil. And that is even close to the upper bound of bacterial load in some of the richest soils. And also viruses can regulate their host population dynamics. So in the figure below here, it shows bacillus RNA counts and their associated phage genome counts in metagenome sequence from biocrust soil, and also clearly shows the nice prey and predation relationships after the soil wetting. And also viruses can influence their host metabolism. So just recently, viruses were found to carry some auxiliary metabolic genes, or AMGs, that can be potentially functional while infecting their host. And here shows an example of AMG detecting on viral genomes assembled from freshwater and marine metagenomes that can potentially oxidize sulfate and sulfate, and will in turn improve the fitness of both the viruses and their host. And also virome is a really complex and largely unclassified community. So for environmental viruses, we're talking about here, we're mention about the whole community rather than certain or one types of deadly pathogenic viruses. So the whole viral community is extremely diverse, but the fun part is that there are lots of unknowns. So here shows an example of a gut virome, which should be relatively better characterized systems compared to soil. And each nodes in this Bayesian network represent a detected viral genomes, and only the nodes that highlighted in red were classified. So we still do not know much about virome within a system such as gut. Then the next question is like, how can we decode these mysterious viral communities within the given system? So in the recent perspective paper, Ru et al. Outlined the main method that have been currently applied. And these methods could be generally classified into methods of detecting free viruses, method detecting cell-associated viruses, and method of potentially detecting both. So for methods of detecting free viruses, so the most straightforward way that you can imagine is to isolate these viruses if we can, and then sequence them. But if we cannot, we can first enrich those viral fractions and then sequence them. So we often call the metagenomic data of these enriched viral fraction by Rho-mix. And if you're interested in this viral isolation or enrichment from soil, I would highly recommend you referring to the protocol that published by Joy Emerson's team and Gary Trubel. And here's our titles of those protocols for your information. And then if you want to look at the cell-associated viruses rather than a whole community, then you can either like isolate the cells and sequence them, or assemble the microbial genomes from the metagenomes and then recover the associated virus sequences if there's any. But if you want to study the whole viral community, including both free and cell-associated viruses, then screening viral signals from metagenomes could be an option. So as bulk metagenomes is the main topic of this summer school this year, and here in this talk, I will mainly talk about the approach of screening viral signals from bulk metagenomes. And it's also important to note which method to use, what type of viruses to detect should be tailored according to your specific study aims and hypothesis to test. Therefore, there are several things that need to keep in mind before applying this method to your study. So as mentioned, this method could potentially capture both free and cell-associated DNA viruses with relatively less sample preparation needed, such as isolation or enrichment. And there are more bulk metagenomes currently now available compared to viromics if you want to do some meta-analysis. But the disadvantages are the low coverages, and also the data could be relatively noisy when you're screening the virus signals. And also these are reasons that will contribute to some of the challenges that we're going to talk about later. Then assuming we have the metagenomic data on hand now, and then we'll need to be clear about what viral information we want to get out of it. Then we need to write down our wish list or specify our objectives. So for any microbial community study, there are two keywords, structure and metabolic function. And that is also true for viruses. So deciphering the structure and metabolic functions of viral community will correspond to the first three objectives listed here. So first, we need to screen the virus sequences from metagenome assemblies, then classifying the virus sequences, and also functional annotating the genes that caught from the virus sequences. In addition to those, as viruses also depend on its host, then there is an extra objective for viral communities is to identify their host communities. So now we have this clear objectives. The very next step is to select methods or tools that can help us to achieve those four objectives. Then for the first step of screening viral context from metagenome assemblies, I'll list three most widely used tools with numbers of citations for each. So here we have virus sorter by Brand, virus finder, and also we have another tool called CheckV that is used for evaluating the quality of the detected viral context. So both virus sorter and virus finder now have a new version. Here we refer them as virus sorter 1, virus sorter 2, virus finder, and deep virus finder respectively. So all these listed tools are relatively easy to install with detailed documentations on their homepage. And also you can install them with Conda. So you do not need to worry about installing the dependencies or worry about the dependencies are not compatible to each other if you install the tools in a specific Conda environment. But if you have not used Conda before, I will highly recommend you checking it out. So now we have several candidate tools to use. Then the next is to understand the methodology they applied and the supporting theories. So by understanding those, it will help you to select tools that are more suitable for your own study. So in general, the method implemented by these tools for this particular step can be categorized into two types. So one is mainly based on the sequence similarities to the references and complemented with the scoring metrics. The example of this general type of method is virus sorter 1. So virus sorter 1 will first detect significant similarities with known virus sequences in NCBI and also environmental viral data. And then also screen for viral-like genome structures, such as like depletion of non-viral genes or enrichments in these short genes. So these results will also be put into primary and secondary metrics and build probabilistic models to evaluate if a given sequence is more viral-like or is just expected by chance alone based on the overall frequencies of viral-like genes in the whole data sets. Then the second general type of method of the step 1 is the hybrid of viral sequence signatures and machine learning. So the features used for machine learning model here could be either annotation metrics, such as what is used in VirusThought 2 and Vibrant, although they're using different annotation databases. And alternatively, the nucleotide patterns of the viral sequences that is distinct from the non-viral sequences can also be used as features that is implemented in the Viral Finder and Deep Viral Finder. So by applying this machine learning method, we could possibly explore more diverse viral sequences beyond the database representation. Then the different tools that use machine learning listed here can access different viral features or signatures to retrieve more viral sequences from the metagenome assemblies. And all these tools I mentioned are widely used and well-recognized tools for screening viral contigs from metagenome assemblies. But sometimes more is not always better. So here shows a Venn diagram that is the numbers of viral contigs identified by VirusThought, Vibrant, and Deep Viral Finder from the same metagenomes that I'm currently analyzing. And it clearly shows that there's some consensus in between the different tools. But there also there are some sequences that were detected as viruses by only one tool. So this is, and also I could check the numbers. So this represents how many of those putative viral sequences are considered with high to medium quality. So these differences in viral identification result from the different tools could be due to the different database they use or different viral features they assessed that can result into different sensitivities or positive discovery rate when detecting the different types of viruses. And also these differences were observed in another study when comparing the F1 score. So F1 score, we evaluate both recall rate and precision when comparing different tools for detecting REF-seq double-strand DNA viruses and also non-REF-seq double-strand DNA phages and also the prophages with different length. So in addition to these differences, another remaining challenges is that we're limited to the numbers of reference viral genomes, as both type of methods rely on the database, no matter it's for annotating genes from putative viral contigs or for modeling the genomic features of known viruses. And also in the same perspective, articles by Ruonan et al. I just mentioned, it says that uncultivated viral genomes have been screened from the genomic data sets nearly 5 times the total numbers of genomes screened from viral isolates. And also these uncultivated viral genomes represent more than 95% of the detected taxonomic diversity in the public available viral sequences. So that means now we're using the 5% to capture the rest of 95% of viral diversities. And also these numbers could be constantly changed when there's more viral data become available. Then another remaining challenges is that to determine the completeness of the viral genomes. So unlike the bacterial genomes, viruses do not have a set of universal marker genes. And also their genome size can vary a lot, ranging from 1 kb to several MB. And also their genomes can be either circular or linear, and their genomic contents can be highly diverse. So the current method that used by CheckV is based on, so the completeness is based on a comparison with complete viral genomes from NCBI gene bank and also environmental samples using average nucleotide identity and percent of coverage. And also their closed genomes were identified based on detecting either direct terminal repeats or flanking host regions on proviruses or inverted terminal repeats. So hopefully in the future, we can learn more feature coded in viral genomes that can help us to better improve these methods. So now hopefully we have some idea, like understand the basic methodology that applied to these tools in this first step, and also some remaining challenges. And here are some of my little suggestions. So it will be helpful to run at least 2 of these identification tools and integrate the result to improve the detection confidence. So in our recent study, we integrated tools that applied complementary methods and set 4 customized criteria and only keep the context that satisfy at least 3 out of 4 criteria. And if you're interested of this type of workflow or methods, so feel free to check it out. And also another little suggestion is that when we explain the result, it's better to report on the positive findings and speculate less on the negative result, as not detecting a virus in the metagenome does not mean it is not present. Also, another reminding is that it's always important to compare results across studies that apply the same method. Otherwise, the result could be misleading. Okay, I think we come to step 2. So now we have screened the viral context from the metagenome assemblies. Now the next step is similar to the applicant data analysis, if you're more familiar with, that is to cluster these viral contexts or viral sequences to either evaluate their diversity or compare across samples or assign taxonomy if they are clustered with some reference viral genomes. Then in general, there are 2 widely used methods for this step. So the first one is based on the whole sequence alignment at kind of 95% of coverage, 95% of average nucleotide identity, over 85% of alignment fraction to cluster viruses at species level. So when Ruonan et al. Suggest those cutoffs, they first compare the NCBI RAFSIC viral genomes and found those cutoffs could better capture the clusters of viruses and suggest to name these clusters VOTU, or Virus Operational Taxonomic Unit. So that is used to differentiate from viral population or viral clusters that are used in other literatures where the different clustering methods were applied. And also the documentations of these computing steps and also detailed scripts can be found where this link, or you could simply search the repo name as shown here in the GitHub website. So this script have been published for literatures studying DNA viruses from human gut microbiome, but the methodology used here are the same if you want to study soy viruses or marine viruses. And alternatively, you can use other tools or scripts that you feel more comfortable to use, as long as you can calculate the average nucleotide identity, alignment fraction, and applied clustering algorithms. So as long as you understand the fundamental methodology and a consensus reached on the selected cutoffs, then the detailed steps can be relatively flexible. So then a second widely used method is based on the protein sharing matrix. And this method have been implemented in a tool called vContact, and now it has a version 2. So instead of the whole sequences, vContact will first cluster proteins that are called from the viral context, and then converted the presence or absence of the protein clusters into similarity matrix among the sequences. Then the protein sharing matrix is visualizing in a network with each node representing viral genomes or viral contexts. And then the network is further examined to identify if there's any outlier nodes or there are clusters with overlapping nodes, or there are complex clusters that can be potentially separate into subclusters. So after this network optimization, then the viral clusters can be used to make taxonomic assignment of viral context at genus level. So it has been tested on NCBI RAFSIC viruses and reported nearly identical taxonomy assignment from ICTV, that's short for International Committee on Taxonomy of Viruses. So the vContact also include reference viral genomes that you can cluster together with, and also the team is constantly updating the reference network when more viral genomes are becoming available. So vContact 2 is also really easy to install with Conda, and also available at Kbase and Cybus as an app, so it's pretty handy to use. I just recently have seen another tool called Demover to classify viral context to order or family taxonomic level by comparing genes on the amino acid levels against the viral subset of TR-AMBL database when taking a vote on of the order or family hits. it is also important to note that these UniProt KB TR AMBL database contains the unreviewed entries that are previously intentionally kept separate from the UniProt KB Swiss Prot database that contains manually reviewed entries. But it's also good to take advantages of exploding data, as always. But I haven't compared these tools with the others, but this method seems like cited in many recent papers and review articles. So it should be another interesting tool to check it out as well. Okay, then here comes the challenging part again. So both the whole sequence alignment method or method 1 or protein sharing method or method 2 relies on the assembled contigs or genomes that are long enough to share significant overlaps with others so they can be clustered together. So, but the assemblies in the metagenomes are often incomplete and short in length. So then the partial genomes are still remain challenging to classify. And also in addition, even for the ones that are long enough, the genomes of environmental viruses are highly diverse as mentioned. And here is a similar heat map, but those are for the viral sequences that deposit in the AMGVR, a database with higher representation of the uncultivated viruses detected from genomic data. So the same cutoff are still informative to some extent, but do not cluster the sequences, the viral sequences that we have seen in the previous NCBI REF6 viruses as clean clusters in that case. So it's still, when it's applied to environmental viruses, it's still some problems there. So here I quote what is mentioned by Ru et al. That is always good. A good practice to report the clustering method, the cutoffs, the reference database that are used, and also the genome alignments approach, because small differences have been observed between different methods. Then here, another suggestion is that one needed. It is also good to integrate different tools with complementary method. So for example, we previously integrate the protein sharing matrix and the tetranucleotide frequency patterns to cluster viral sequences identified from soil metagenomes assemblies. Then one more suggestion is that we should select suitable methods based on the needs of your own study. For example, if you want to compare results with other studies, then applying the same method is critical. And if you want to merely compare samples with your own study, then it is more important to apply consistent method or criteria that fit your own data. Okay, then step 3, the next step is the functional annotation of the viral genes. So recently there is an increasing interest in detecting cellular metabolic genes or AMGs. So AMGs are genes found in viruses and originated in microbial cells and not directly needed for viral replication, but can modulate the host cell metabolism during infection and potentially in turn benefit the viral replication or survival later. Then examples of those AMGs are like photosynthesis-related genes carried by cyanophages, the viruses infecting cyanobacteria. So from the definition of the AMGs, you probably already know the 2 main steps of detecting AMGs carried by viruses. So first is that we need to make sure the genes candidates are from the viral region rather than the linked host genomes. So Vibrant and DRAM-V here are the tools that are widely used for detecting AMGs. So Vibrant will train the non-viral regions before searching for the putative AMGs to make sure the genes are from our viral genes. Then DRAM-V will only take the contigs that are identified as viral sequences without contaminations of host genomes from viral fodder output, and also carefully examine the gene arrangement along the viral contigs to make sure the putative AMGs have viral genes both up and downstream. Then the second step of detecting putative AMGs is to screen the genes that are involved in microbial metabolic pathway. So for example, in DRAM-V, it will remove some of the pathways that are related to host cell attachment or cell entry, So that doesn't seem like a metabolic pathway of to viruses. So both of these tools are really nice one to identify AMGs and also really easy to install with Conda. And also they're available at either Sybase or Kbase as app. Okay, challenging part again. So there's still some limitations and challenges remaining to detect other metabolic functions carried by viruses. So one challenge is at the functional annotation steps. So from my own experiences, so sometimes nearly half of the genes are caught from the viral genomes are hypothetical. So we're still leaving many of these viral genes unclassified. So our knowledge of viral genomics is still lacking. Then another limitation is that the functional annotations of putative AMGs is only an indication of metabolic potential, with limited information of whether the viral genes can encode a protein, and this protein can fold into a functional enzymes, and this enzyme can be expressed and catalyze reactions as we predicted. So there are currently several soil viral study report the detections of AMGs, and only one were detected from salt permafrost soil were expressed and also confirmed to be active in cleaving mannels or plant-derived polymers. So the majority of the reported AMGs are bioinformatically annotated and assigned with functional potentials based on their similarities to microbi databases. So this approach definitely overlooks some critical motif or catalytic site that are really key to determining if a protein is functional. So therefore, obtaining a protein structure could provide an opportunity to gain more insight on functions of AMGs. Then the protein structures, it's ideally obtained through some imaging, for example, the crystallography. And for example, in our recent study in collaboration with JGI and also SLAC, so we expressed and purified and crystallized soil viral chitosanase and solved protein structures and validated its enzymatic function and also the function of the active sites, and also demonstrate how the proteins could bind or interact with the substrate. But if the conditions not allowed, then it could be also informative to model the structure. For example, using the AlphaFold tool, now is the open source software, and also currently installed and available on EMSL HPCC. But if your team are not ready for all of those, then we need to be careful when freezing the result to avoid overstating the findings. Okay, then finally we came to the last step of predicting host of identified viruses. So the currently available tools can be categorized into three general types of method: alignment-dependent methods, alignment-free methods, and integrative methods. So I also append the numbers of citations of each for your information that may facilitate you to select tools to use. So alignment-based method or dependent method, mainly based on the CRISPR spacer matching or sequence similarities to the reference viral genomes with known host, or the prophage regions of metagenome assembled genomes. Then alignment-free method are primarily based on the oligonucleotide frequency patterns with the probabilistic method, or extracting sequence features of known viral host pairs to construct the predictive models. Then integrative methods are the ones that combine multiple features that I just mentioned. And for example, the viral host matcher net integrate both the CRISPR spacer matching method and alignment-free similarity measures. So I have run some of those highly cited tools, such as the viral host matcher, WISH, PHP, and also some of the tools that retrieve and match the CRISPR spacer with no problem installing and running those tools by following their instructions on the homepage or GitHub page. And those maybe highly cited tools could be your first try if you want to explore data to predict the host of your identified virus sequences. Okay, then here I list some of the challenges and limitations of the current method for the step 4 here. So first up here is that the alignment-based method, such as the one that search for the sequence similarities to the reference viruses with known hosts, for example, the VFP class method, are highly dependent on the reference database, and then may potentially limit us in predicting host viruses that are not well represented in the database. Then another limitation is for those methods of matching the CRISPR spacer, because it will only able to detect hosts that can encode the CRISPR-Cas systems. And also, additionally, as the CRISPR array, as you know, is a kind of history record of the previously conjured invaders. And also the length of the CRISPR array depends on the ability of the host to maintain the systems over time. So therefore, the CRISPR spacer method, matching method, may not reflect the active infections between the virus and host pairs at the times of sampling. Then the last challenge is that I would like to highlight is the problem of false positive, as always. The figure here shows a comparison between different methods. Some are like alignment-based and some are like alignment-free methods. So in the panel A here, it shows the predictive accuracy were increased with the taxonomy levels from the genus to the phylum and definitely domain. So those tools do a relatively good job on predicting host phyla, but when it comes to the order or lower level, the accuracies of some of the alignment-free methods were lower than 0.5. But the CRISPR spacer based method that predict the viral host, which in the darker green bar, can predict the host for in a pretty good accuracy along all the taxonomic levels ranging from 0.77, if you can see, and to 1. However, it only predict the host for less than 1/4 of the viruses, as shown in the panel B about the recall rate. So this comparison suggests that the tool used different methods could be used in a complementary way. So I would suggest to run tools that implemented with different method and apply consensus approach to post-process your host prediction results. Okay, then hopefully now you have a general idea of what basic viral information we can get from the metagenomes. And also you have a list of tools for each step that you want to give a try. And also keep in mind there's some potential challenges or limitations with my latest suggestions on how to work around or mitigate them. Then in the perfect world, we'll have clean viral data screened from this noisy metagenomes with step 1. And we can group those identified viruses into little boxes, and hopefully we have label for each of them, the bacteriophages, the giant viruses, etc. We're step 2. And if there's any exotic metabolic genes that carried by your identified viruses, then we'll get them and screened and classified in the step 3. And also in the step 4, and hopefully we can gain some insight on what microbial community that viruses in your system of interest are potentially influencing. Then at last, there are several tips or notes for you when we are doing the bioinformatics. So there are always new tools emerging. I will try to catch them up, but yeah, they're fast. But the understanding the principles and differences in methodology matters. And I put an expansion mark there. And also the tools that can help you to answer your specific scientific questions are the right ones to choose. And I think that is the end of my talk. And thank you for attention. And I would like to answer any questions you may have. Thanks.
Ruonan, that was great. Thanks so much. I'm going to check the Discord here. Okay. So there was one relatively quick question from Alice. There's a reference on slide 2, Ruonan, Gregory 2020. Maybe when you get a chance at some point today, if you could just post that reference into the Discord.
Sure, do it right now.
Great. And then maybe while kind of folks are taking all of that in and thinking about what questions they want to ask, I can just get things rolling. So you and I, Ruonan, have worked together a lot on this. I'm just kind of wondering, like, when you get all of this data in for either metagenomics or viral metagenomics, the networks that you're showing, the heat maps, these kind of maps of the genomes, they're very complex, and sometimes they're very hard to interpret and visualize by just a person looking at them, whether that's on a slide or in a publication. So, like, just kind of keeping with the theme of visualization and statistical analysis, like what are your preferred ways to kind of generally take all of these large complex data sets, and kind of what tools and what kind of methods do you like to use to kind of compress that down into something that a human being can actually look at and interpret?
Yeah. So that is often a question that we're asking ourselves. Oh, that's kind of a complex network, and how can we retrieve informative result out of it? Like as mentioned, it really depends on your study aims and objectives and scientific questions you're asking. And just for example, that we're currently working on some of the virus host infection network. One is response to differences in soil moisture. So when we see some clusters that is particularly interested to us and is like, oh, this potentially viral generous there, then we'll probably put out that clusters and look into details which is linked to which and what their genomic underlying mechanisms there might be. So it really depends on the questions and your interests and how. So the data always serve as a pool that you can retrieve information that answer your own questions. So as long as it's kind of backwards way of thinking. So
yeah, no, that's great. Yeah, and this is, I think, a common issue with these kinds of data sets, and it's a good opportunity, I think, for an intersection between data scientists like ourselves and maybe folks who have more expertise in visualization and getting these messages to people in the best way. I think I've had some really good success talking to graphic designers and artists and things like that to try to make figures that convey the most information with the least amount of confusion. There was another question here from Paula. So moving away maybe a little bit from metagenomics and metabolomics, but any tips for analysis of viral signals from metaproteomes from soil?
Yeah, talking about metaproteomes, we also have a paper that published in 2021 that integrate the omics, metagenomics, and metatranscriptomics and metaproteomes to study the soil viruses. So welcome to check it out. I can also put that link in the Discord chat.
Yeah, I think that would be great. So, when you're thinking about analyzing viruses from a metagenomic point of view, which is kind of especially when you're talking about AMGs, their potential to contribute to the system, because the gene is just a potential for something happening, versus the metaproteome, which is much closer to their actual expressed phenotype. Are there any differences in the way we should think about interpreting that data, things that they can do versus things that they actually are doing?
Yeah, so like you mentioned, the multi-omic integration should gain some insight on that. Like the transcriptome can be maybe transcriptional levels, and proteins they already have this kind of capsid or some enzymes already there, the kind of indications in that as well. And also I know some when doing isotope or something in other groups, so that could be another thing. And also viral tag to label your virus of interest and track along the time and do the multi-omic. So there's so we already have these tools developed, not we, but in the research field. We just need to think how to apply these tools in the virome study. So yeah. So
great. Maybe just while folks are thinking, I'll take the opportunity to ask one more. And please, everybody go to the Discord channel and put in your questions there. Even if you think of a question after we're done here, we're going to keep monitoring this, and we can certainly keep trying to answer questions even after the talks are over. So please post your things there. So Ruonan, one other question I had was kind of looking at this AMG analysis. What do you think the differences in the viral life cycle might have as far as whether an AMG is proliferating or being used or not? Because if an AMG is being added to the bacterial cell by an incoming virus, but then that cell immediately replicates and lyses the bacteria, then how does that change maybe the dispersion of an AMG versus when a virus is more lysogenic and inserts into the genome, and then through replication it gets further and further? How should we think about that when we're thinking about how AMGs can disperse?
Yeah, and I think it really depends on the environmental context itself. So in marine, they said, oh, the viruses like in the driver's seat, like they're really selfish, replicating themselves as long as they can get into the cells and then bring something to trick the whole cells and get them grow and then burst them. And so selfishly benefit themselves. But also in soil, I think there is a recent paper on some soil contaminated by a heavy metal, and they found there is a lot of prophages, meaning that's lysogenic stage, and carry the AMGs that about these heavy metal mitigation or something that can make them the host or like can resistant to this kind of metal contamination. So this kind of like dynamics like environment, and also it really depends on the environmental context, like how, I won't say like viruses decide how the life cycle will change dispersal or expression of the AMGs, but it really depends on the environment itself.
Great. So, okay, well, we're getting close to the hour when we will all get a well-deserved break.